everyone. Um, yeah, so I did have strep throat. My throat is not 100%. My fever just broke yesterday. So I have been very sick this last week, so I do apologize. Um, so no, I did not have COVID. I've heard that there's been a couple of rumors. People thought I was really sick in the hospital with COVID or something. No, it's not true. Um, I had a really, really bad bout of strep throat. So yeah. Uh, so I do apologize if I sound a little quiet or if I take a little break from talking. Um, my throat is really, really sore. So I'll do my best to get through everything. Okay. But we'll start off with chapter 6 where we left off. I believe it was here, ATP synthase. All right. So I think we covered here the electron transport chain. The electron transport chain is going to be utilized again to harvest it's going to produce an electrical chemical gradient, which will then be used to harvest ATP. Okay, so this potential, this electrical chemical potential, and I don't know if we went through, or did we end off here? Let's start early on. Let's start glycolysis. Okay, I think that's what we did. Yeah, we'll start here. So again, glycolysis, first part of this series of energy harvesting metabolic pathways. We break down glucose in order to gain this pyruvic acid, but most importantly is to gain to ATP. Okay. I think I already covered glycolysis, but let's just start here. First, in glycolysis, you have to invest 2 ATP or 2 units of energy. This ATP will then be used to phosphorylate the ends of this glucose that makes this molecule unstable, and it actually allows this molecule to break down into 3 carbon sugars. NAD comes to remove electrons. This is important because this allows for the harvesting of ATP. Okay. Again, you need to invest energy in order to get energy from glycolysis. Also, the net gain of ATP in glycolysis is 2. Okay, so it's very little. This can function without the citric acid cycle and without the electron transport chain and without the ATP synthase. Okay, so this can happen without the big energy harvesting steps. Okay, and that's kind of important to note because this functioning without the other stuff means that you can perform this without the other stuff working. Okay, and this is what organisms do without the presence of oxygen and without the presence of the terminal electron acceptor, which we'll talk about in a minute. So this is separate. This can happen with or without oxygen. So this is what will occur if you guys are running sprints and you have something called anaerobic conditions in the muscle tissue, and you have lactic acid fermentation, right? So this is when let's say you're in high school and you guys are playing sports, or you have a really hard workout, and you sprint, or you're running, or you're lifting heavy weights, and your muscles function without the presence of oxygen. Instead of pyruvic acid, you can have a buildup of something called lactic acid. Okay, and this is going to be a fermentative route. All right. So just remember, glycolysis is separate than these mitochondrial functions. Okay, so the citric acid cycle, which is also important. The whole point of the citric acid cycle is to get these NADHs. Okay, this is changing NAD to NADH. Right, NADH will then be used to dump the electrons off to the electron transport chain. which will then be allowed to pump hydrogens out of that inner membrane or outer membrane of the mitochondria to make that electrical chemical gradient. Okay. So the pyruvic acid again gets broken down one more time, and you get this acetic acid, which can then plug in with the help of coenzyme A into this process of the citric acid cycle. Okay. I thought this graphic was confusing. Okay, so I do remember this. We were here last time. I believe this is a better representation of the process. Okay, so one full revolution around this metabolic pathway is half a glucose. Two full turns around this process is actually a full glucose molecule. Okay, because it's only going to be that 2-carbon plug-in from that initial pyruvic acid. So 2 acetates will plug in to make a full glucose, if that makes sense. You lose CO2 and you gain a lot of these NADHs, okay? In terms of energy, this is very inefficient. This is not an energetic metabolic step, okay? You only gain 2 ATP. So this is just as efficient as glucose in terms of making energy, but you waste all of these NADHs, right? So remember we talked about last time, well, it's been about a week now, those NADs that we need to have the NADs free to rip off the electrons in order to harvest the energy. Oops, what is that? So you need to have these NADs available to harvest the ATP here in glycolysis, okay? If this NADH is present in the body at high concentrations, it's not going to be efficient. It's not going to push forward this movement. And again, glycolysis only requires 2 NADs, okay? Whereas look how many NADs one glucose molecule needs in the Krebs cycle, right? You need 6 and 2 FADs, which is a lot, okay? So just think about that in terms of the efficiency here, right? You only have the ability to harvest 2 ATPs. You gain a lot of NADHs and 2 FADs, okay? And you lose, of course, the 4 CO2s or the carbon dioxides, okay? So again, very inefficient in terms of gaining energy, right? Has the same energy gaining potential as glycolysis, but you're getting a lot of these NADHs. And again, these are going to be important for the citric or the electron transport chain. Okay, so this again is only important. This is pretty much only useful for gaining these NADHs and these FAD H2s to plug into the electron transport chain. Okay, so I do want you to know these total amounts here and why this is important. And again, the majority of this is what we call reducing power, or the ability to donate these electrons to a different metabolic pathway, okay? All right, so now we're gonna go and talk about the electron transport chain. Again, this is where all those NADHs are being dumped onto or plugged into. Excuse me. And again, this is what's gonna create that electrical chemical gradient or that electrical chemical potential. All right, so here we have these NADHs. This is kind of a little graphic showing how these electrons are going to be working. You have this this power up of these electrons, or this potential of electrons that are going to be dumped down into this pathway. Again, this electron transport chain is going to be used to kind of power these different enzymes in order to pump hydrogens out of the cell and then eventually produce a proton motive force to work a enzyme, which I'll talk about later. It's going to be called the ATP synthase. I think there's a better, yeah, that's probably the best graphic right there. But essentially what happens is you have these NADHs that come and the NADs come, they rip the electrons off of the different glucose molecules or the different glucose molecule pathways, um, and they will dump their electrons onto this electron transport chain. So what happens? Oxygen is going to be utilized in this pathway in order to have an attractive force that'll pull these electrons in this pathway. You have all of these enzymes here, these cytochromes that are going to be passing these electrons off. And during this pass of electrons, you have the pumping of protons out of the inner membrane of that mitochondria. Okay, and what happens is this acidifies the environment, and this creates that potential. Okay, and when I'm talking about potential, I'm talking about almost like a barrier with differences in concentration. Okay, and in terms of the differences in concentration, right? You can have like a potential, like a dam, where you have a barrier and a water potential where the water will want to rush down through the dam in order to make that energy, right? Well, here we're going to have a potential of protons, okay? We'll talk about AT synthase right now. But I feel like this is probably the best graphic for that. But this is the electron transport chain, right? This is where the NADH comes and dumps off the electrons, right? So as these electrons are dumped off, you have these enzymes, these electron carrier complexes that are going to have these electrons and pass them off. Cytochromes will pass these electrons off to the other embedded membrane proteins. When you dump the electrons here, they remove a hydrogen from inside the cell and pump them outside, right? So this is going to be acidic. Is going to make sure that there is space, okay? Uh-oh, it says my internet stable is not strong. Let me know if anything happens. I'll come back on if it drops, okay? But again, these protons or this acidified environment is going to provide a force, okay? And this force can then be utilized like a dam to harvest hydrogen or harvest ATP through the hydrogen or proton motive force, okay? Through ATP synthase, all right? And that's pretty much what you need to know about the NADHs, okay? They're gonna come and dump their electrons off on these enzymes, and they're gonna be pumping these hydrogens out, all right? So on the exam, I might provide like a graphic, and I might ask you what these enzymes are doing, and you could just say they're pumping the protons out to make a gradient, right? In order to have this enzyme work, okay? And I'll explain this enzyme right now. But just know that these electrons are going to be utilized to make a gradient to then harvest energy, okay? All right. So ATP synthase, again, is a protein cluster found in a cellular membrane, including the inner membrane of mitochondria, the thylakoid membrane of chloroplasts, and the plasma membrane of prokaryotes, that uses the energy of a hydrogen ion concentration gradient to make ATP from ADP. And ATP synthase provides a port through which hydrogen ion concentration gradient to make, oops, I think that's a double one. Yep, I wrote that twice. So again, so it's just reiterating ATP from adenosine diphosphate. And ATP synthase provides a port through which hydrogen ions can diffuse through, okay? So think of this as the dam that's going to be, the component of the dam that's going to be harvesting the electricity from water, okay? So how a dam works, if you don't know, is there is a wall, again, holding back the water, that potential energy, that water that's going to be pushed back, right? The dam works by letting the water out. And when that water gets let out, it's going to be turning a turbine as it passes through that dam. And this turbine is going to be turning, and then that turning of that turbine is going to be used to harvest electricity from, okay? So the water rushes through, turns the turbine is going and also creating electricity, okay? So ATP synthase works almost the exact same way, right? This arrow is referring to the turning of this enzyme component, and then harvesting of energy that way. Okay. Um, so what happens is you have this potential here, and this potential, instead of water, it's going to be hydrogen molecules, right? So here we have less acidic uh, um, uh, amounts of protons here, and more acidity here. So we have a potential, right? Also, this is much more positive than inside the cell or inside of this space. Oh, excuse me. Okay, so you have a much higher positive charge on the outside, and as well as a much higher acidity concentration on the outside, so a lower pH here. Okay, this protein is going to allow for the passage of the protons to come through, right? Because this barrier isn't going to allow that. So this passage of this hydrogens is going to turn this enzyme turbine. Okay, it's going to turn, it's going to allow for this to be harvested and convert ADP into ATP. So again, ATP synthase, right? It's going to synthesize these adenosine triphosphates. Okay. All right. If you have any questions, please let me know. I know this is kind of confusing. Again, I'm giving you the detail behind this, but there's a few things that I do want you to know only. This potential gradient here that we use to harvest ATP from. These membrane proteins that are going to be utilizing the electrons from NADH and FADH2 to pump the protons out, okay? And then just know that this is also going to be the two main components that happen in the mitochondria, right? So glycolysis is going to be a different process entirely. It can happen separately. Okay, so here's a summary of ATP yielding processes in the cell, right? You have glycolytic pathway, which is going to be separate from the mitochondrial steps, right? So this can occur with or without mitochondria, right? There's certain organisms, eukaryotic organisms, that don't have these steps here, okay? They're going to be simple organisms like Giardia, or parasitic organisms like Giardia, okay, that require like hosts to survive. But again, they do have or lack mitochondria. Okay. Again, citric acid cycle is only used to gain pretty much these NADHs and these FADHs to go down this electron transport chain. Okay. This is only going to yield to ATP, which is the same as glycolysis, but you only take up those two NADHs, right? You need this NAD again to convert into NADH for glycolysis, or else glycolysis does not work. Again, the majority of our ATP is made through the ATP synthase, which is 28 ATPs, which is way more efficient than glycolysis itself. Okay, so just know that as well. But the mitochondria being present and utilized is going to give you the majority of your ATP. All right, so here's a nice little graphic kind of showing you how different types of foods can be converted into energy. You can have carbs, which are kind of the easiest source of converting into glycolysis. Glycerol from fats can eventually convert or go into the glycolytic pathway. Glycerol is a 3-carbon sugar, which can plug into the glycolytic pathway here. Oops, I think I went too far. Yeah, so 3-carbon sugars can plug in here, right? Instead of having the breakdown and separation of the 6-carbon sugar, you can start right here with the 3-carbon, okay? And then you can also have the breakdown of protein. You can either start in glycolysis, converting some of these amino acids into a sugar component, or you can plug into the citric acid cycle, okay, acetyl-CoA. So this will just require a 2-carbon molecule, if you remember, right? acetic acid. A compound that looks like acetic acid will be fine. Okay, so you can plug in at different parts of the pathway in order to gain energy throughout these different different parts. Okay, so again, this is just showing you that you can gain energy from carbohydrates, fats, or proteins. Right? They just might plug in to different parts or different metabolic start points or entry points, right? So glycolysis is the start or the beginning of it all, but again, you can plug in before the citric acid cycle, during the citric acid cycle, in order to gain your energy. Okay. So fermentation, we talked a little bit about fermentation already. I talked about you being able to ferment if oxygen is not present, or if you're working out too vigorously, you have the lactic acid fermentation, right? So anaerobic means lacking or not required molecular oxygen or O2. Fermentation is the anaerobic harvest of energy from food by some cells. Different pathways of fermentation can produce different end products, including ethanol or lactic acid, right? So yeast will ferment, right? And they'll have an ethanol fermentation process, right? This is where we get our alcohol from. Whereas humans or mammals will have lactic acid fermentation, okay? So this is going to be converting that pyruvic acid into lactic acid, again, in order to harvest that NAD, okay? So why do we ferment, okay? So this is the main reason why we ferment, and it's in order to keep the glycolytic pathway or glycolysis working, okay? And why is that important, right? So we need, if you don't remember, we need NAD to remove electrons in order to gain our ATP. So I want to show you this process again, and this is going to be very important. This process right here, step 2, in this 3-step process, right? Energy harvesting phase here. This process here, the NAD to NADH, is going to be what allows us to gain our ATP, okay? And I know that sounds weird, right? Removing of electrons will allow for the harvesting of our ATP, okay? And excuse me, what that means is that if we don't have this process occur, we just donated to ATP to break down glucose, and we're going to lose the molecule. So we don't have free NAD, we don't have a working glycolysis or glycolytic pathway, okay? So we need NAD free in order to gain our electrons, right? So what happens then is when we have the pyruvic acid, we can donate the electrons back onto this pyruvic acid in order to recycle NAD to go back and again remove the electrons off. This excuse me. This process was going to be very important in order to keep glycolysis working. This recycling of the NADs, okay? So you can form these toxic organic molecules in the body, which the body again will deal with eventually. But for fungi or fungus, yeast, they ferment lactose, or they ferment into ethanol instead, right? Their byproduct is ethanol. Ethanol is not good, okay? It might be good, right? We like beer, we like wine, we like liquor, but it is not actually good for the cell. Alcohol is toxic, right? You can use alcohol and sanitizers to kill microorganisms, okay? So the byproducts are toxic. They're not good for the cell. So this process, again, this anaerobic fermentation, is typically not efficient, right? Because the glucose is not working. And you get this toxic byproduct as well. So this is kind of the last resort, the last mechanism in order to keep the cell alive or keep energy flowing, okay? Again, if you do not have oxygen, you will die. You cannot, our bodies cannot subside on a fermentative process, okay? We need to have oxygen in order to survive. And that just shows you that this process can really only occur to produce energy in muscle tissue, or else you could die, okay? Um, so again, yeah, process is only really for the separation or the gaining of these NADH or these NADs back to use energy again. All right, so here's what I was talking about before with yeast. This fermentative process produces ethyl alcohol. Again, we like ethyl alcohol either for drinking or for antiseptic purposes. So their yeast have a little bit of a different pathway. Yeah, we won't talk about this. Now we'll get into photosynthesis with a little bit of time that we have left. All right, so chapter 7, photosynthesis using light to make food. Okay, so photosynthesis, the process by which plants, algae, and some bacteria transform light energy to chemical energy stored in bonds of sugars, right? This process requires an input of carbon dioxide, CO2, and water, H2O, and produces oxygen gas as a waste product, all right? And that last slide that I showed you on that chapter 6 was kind of talking about the concentration of oxygen and carbon dioxide. And this plays a key role in the amount of oxygen that is present in the atmosphere, right? And a key role to how respiring organisms are able to survive on Earth, right? Photosynthesis uses, pulls in the carbon dioxide from the atmosphere in order to make sugars, right? And then on top of that, it pushes out or dumps oxygen as a waste product, which is what we utilize to break down sugars. So not only do plants provide us the sugar, the glucose, they also provide us the oxygen in the atmosphere as well. Okay, this is a very important play in the environment, right? The ability to produce oxygen, the ability to produce sugars using sun or solar energy, again, directly benefits all life on Earth that is a heterotroph. And remember, a heterotrophic life is going to be something that breaks down organic molecules in order to further their metabolism or their ability to survive. Okay, so again, photosynthesis uses light energy for CO2 to harvest CO2 and then convert it into sugars, again, expelling oxygen. So I know I mentioned this before, but nearly all life on Earth can trace its source of energy back to the sun. Okay, so the sun, again, is a keystone energy source in pretty much every single biological system. There's a couple of systems such as deep ocean vents that do not rely on the sun, but again, those environments are fairly rare, and the majority of the surface life on Earth, I think all of the surface life on Earth pretty much subsides or has energy originating from the sun. Okay. All right, so we have a diversity of different photoautotrophs. Again, photoautotrophs are going to be organisms that utilize the sun in order to make their energy, right? We have trees, different types of kelp or algae, right? So this looks like brown algae, like you'd find in Monterey. And then you have different types of cyanobacteria, which again make up a lot. Yeah, it's a little ironic. Kind of sad, but yeah. Um, we'll talk about the fires later. I think we're gonna get into like environmental stuff in California, but the fires probably should have happened throughout the last 30, 40 years, but we've been really good at keeping the fires put down, as well as, everyone's talking about global warming, which we'll talk about later too. But yeah, it's sad. And there's ash everywhere. It's probably not helping my throat out any better either. But so yeah, you have different types of photoautotrophs. So again, the plants, photosynthetic protists, and bacteria. Again, these are going to be very important cyanobacteria in aquatic life as well as for oxygen throughout the world, oxygen concentrations, okay? The oceans pretty much run the show in terms of energy exchanges throughout biological systems, okay? The ocean's massive, has more life than the surface of the earth, right? It's where life began essentially too. All right, so where photosynthesis occurs, and the organelle that photosynthesis occurs in is a chloroplast, right? So just a heads up, when we're talking about chloroplasts and or the organelles associated with photosynthesis, we're mainly talking about protists in plants, okay? That have chloroplasts or chloroplast-like structures. Photosynthetic bacteria do not have chloroplasts. They have pigment, and they have invaginations of their cell membranes. This is where they house their pigment and they can utilize the energy. But again, bacteria or prokaryotes do not have organelles like protists or plants do. Okay, eukaryotes, they will have chloroplasts or chloroplast-like structures. But just to be clear there. So chloroplast, an organelle found in plants and photosynthetic protists, enclosed by two membranes. And a chloroplast absorbs sunlight and uses it to power synthesis of organic foods, sugars. Right, so carbon dioxide into a 6-carbon sugar, glucose. Right. Chlorophyll is going to be a light-absorbing pigment in chloroplast that plays a central role in converting solar energy to chemical energy. Again, we'll talk about chlorophyll A and B, or the pigments that are going to be used to actually absorb the light and produce that energy, or harvest that energy in order to fix the carbon dioxide into a sugar. We have a stoma. This is a pore surrounded by guard cells in the epidermis of a leaf. When stomata are open, CO2 enters the leaf, and water will exit. Oops. And O2 will exit. Sorry, this is supposed to be O. I'll change that right now. A plant conserves water when the stomata are closed. Okay, so this is going to be referring to the gas exchange that needs to occur inside of that cell or that leaf during photosynthesis, right? So there's different types of plants. There's like C4, C3. There's which you won't have to talk about, maybe a little bit when we get into plants. But cactus are very special in that they grow in the desert, right? So those plants will absorb sun during the day, and then they'll close off their stoma during the day as they absorb the energy, and then they'll open their stoma at night when it gets really cool, so that way they don't lose too much water, okay? But there's different mechanisms for how these stomata open and close, depending on the environment that plant is living in. But again, this just talking specific plants have different mechanisms depending on their environment. All right, so stroma. So this is a thick fluid enclosed by the inner membrane of the chloroplast. Sugars are made in the stroma by the enzymes of the Calvin cycle. We'll talk about the Calvin cycle in a minute. So you have a thylakoid, one of a number of disc-shaped membranous sacs inside of a chloroplast. Thylakoid membranes contain chlorophyll, and the enzymes of the light reactions of photosynthesis. A stack of thylakoids is called a granum. Okay, so granum, a stack. Again, this is going to be that single stack of the thylakoid membrane formed. In the chloroplast, grana are the sites where light energy is trapped by chlorophyll and converted to chemical energy during the light reactions of photosynthesis. Okay, so this is just giving you the definitions of each of those components. Okay. All right, so here we have the chloroplast. In the chloroplast, we have the thylakoid membrane. Inside, we have the thylakoid space. each of these stacks of these thylakoids is a granum, and here you have the inner and outer membranes of that chloroplast, right? And they're like stacks. They're like stacks of pancakes. That's how I like to think about a chloroplast, right? Inside those granum, they look like stacks of pancakes, green pancakes. And I was talking about the stomata earlier. These are those pores, right, that allow for the gas exchange. So think of this as almost like your lung tissue, right? You have the breathing in of oxygen and the release of CO2. Here's the opposite. It's the job of the stomata is to pull in CO2 and release oxygen, right? So you can see how this would be a problem for plants in areas where there's not a lot of, or there's very little water, but it's very hot, okay? Um, such as the desert, right? So cactus will actually perform the light reactions and then stop at a certain point. Okay, this is why cactus can grow fairly slow, depending on the time of the year and depending on what types of cactus they are. Okay, or cacti. This gas exchange only occurs at night, right? So it'll absorb all the sun energy it needs during the day for a brief moment, and then these stoma will stay closed. At night, they'll open up, off gas, and then perform the dark reactions or the Calvin cycle, which we'll talk in a minute about, okay? But the light reactions, again, is going to be harvesting the light and producing energy. So it's going to be NADPH and ATP is going to be produced in the light reactions, which we'll see in a minute. So this is the overview of photosynthesis, right? You have CO2 and water. Light energy is going to be then utilized to make glucose, C6H12O6, and then oxygen will be produced. But again, this is going to be the Calvin and the light cycle. All right. So again, you have the light reactions and you have the Calvin cycle. Okay. So here we're talking about the light reactions. Again, this can run separately depending on the type of plant, and depending on the type of environment, right? Because after this off gassing or this exchange, this pulling of CO2, you need to open up the stomata in order to allow for gas exchange, but this will also dehydrate that plant extensively, okay? So cactus or cacti are going to have issues doing this in the middle of the hot desert, right? So they have to separate these two reactions out. One needs to occur at night, the other one needs to occur during the day. All right, so it's important when we talk about photosynthesis to talk about the UV or the electromagnetic spectrum. So this is showing you where light is found in the electromagnetic spectrum, and it's right here, okay? So the middle of the figure expands a thin slice of spectrum that is visible to us. You'll see that on the next slide as different colors of light. So light is present, or light is visible. Visible light is from 380 nanometers, okay, to about 750 nanometers in the wavelength. Okay, so here you have 380, that's 750. And you're going to have different areas where light is absorbed, okay? And one of the cool things about plants is plants are green because plants don't actually absorb this wavelength here of light, right? There's not as much energy that is able to be absorbed with green light, but there are different types of chlorophyll or carotenoids that are going to be used to be excited and actually harvest the energy from, okay? So chlorophyll and other pigments in chloroplast reflect or transmit green light while absorbing other colors, okay? So again, this granum or this thylakoid space is going to be absorbing the light. And this is what's going to excite those different molecules in order to use to harvest NADPH as well as the ATP in order to pull in that carbon dioxide and make sugar in the dark reactions, okay? All right, there's also different pigment other than chlorophyll A and B. We have something called carotenoids. Carotenoids are typically left in many of the plants. They're not as precious as the chlorophyll A and B, right? Chlorophyll A and B have trace elements. Um, these trace elements are typically like magnesium. Those elements are really hard to come by. So plants, what will happen in the fall is they will go into kind of like a dormant stage with photosynthesis again, because it's fall and winter, typically not as much sun or free water that's going to be present. So those plants will pull in those chlorophyll A and B components into the tree and leave the carotenoids out, because the carotenoids are not as precious of a pigment as those chlorophyll molecules are. Okay, and then you'll see these beautiful colors in the fall. Okay, so again, these are carotenoids. These are different types of pigments that will be utilized by that plant or that tree to harvest light energy. But again, they're left in the leaves because they're not as important as the magnesium-based chlorophylls are. Okay, so here we have chlorophyll A and B, right? They're different spectrums of green, right? And then the carotenoids are going to be that orange or yellow color. Okay, and this kind of shows you what range of light they're absorbing, right? Chlorophyll A is getting more on the violet side. Excuse me. Chlorophyll B is getting still in the violet, but blue, and the carotenoids are kind of in between. You get a little bit of blue, a little bit of violet, and then you do have a secondary absorbance pattern here, which is going to be that orange and red light. This is important, right? To be able to have very different absorbance patterns, right? You can still absorb red and orange light as well as the violet and blue light. So just to point out that blue and violet light is much stronger. It's much more, there's much more energy present in violet and blue light than there is in red and yellow light. Okay. But to say that, that's not to say that light is not useful. Red light can penetrate much deeper um, in the canopy, or much deeper um, than the violet or blue light. Okay, and that's kind of important too, right? You want to be able to absorb these wavelengths of light, because sometimes the blue and violet light might not always be available. Let's say if this plant or this tree, this organism is growing underneath other larger plants. You want to be able to absorb and utilize the red spectrum of light as well, just because the red spectrum will be able to penetrate deeper into that canopy or deeper into areas of overgrowth. Okay, whereas blue and red, and then blue and violet again are going to contain more energy, but they might also be problematic to get to it, because there's going to be other plants in that environment that are striving to gain these wavelengths. Okay, because again, way more energy associated with the violet and blue. Okay, think of ultraviolet radiation, right? UV radiation is strong enough to damage DNA. So it's going to have more energy for sure. Okay. Whereas red light, we really don't worry too much about red light. We know it does penetrate deeper into tissues, but it has way less energy. So it's not as bad. Okay. All right. So what happens in the presence of light? That this chlorophyll molecule will absorb light, and typically at the center of this chlorophyll molecule, A or B, you have a metal component, or a metal ion. And this metal ion will have the ability to donate or have an electron become excited. Okay, so light energy will come. This metal ion in the middle of that chlorophyll molecule will vibrate. At a certain point, the vibration, it'll release or emit an electron in an excited state. Okay, so this electron will eventually fall to the ground state, releasing heat and a fluorescence, okay, or light. Okay, and this is what's going to be used to power this photosynthesis, this energy that's needed to harvest the NADPH as well as the ATP. Okay. So again, here we have clusters of these chloroplasts, again, embedded on the membrane proteins. But what you have is this photon of light hitting, and then you have the bouncing of the electrons between these different pigments or these different photosystems. And then you have this electron being transferred up into the primary electron acceptor, right? So this is going to be reaction center of chlorophyll A. This reaction center, again, is going to be transferring these electrons up to an electron acceptor, okay? This electron acceptor will then be utilized either to have a proton mode of force, or you will have the electrons being donated as building blocks to NADPH. All right, I think we'll stop here. Let's see. Like 5 minutes. We'll go through this. Yeah, we'll just talk about these 2 slides real quick. So how light reactions generate, excuse me, ATP and NADPH. So photons excite electrons in the chlorophyll of the first photosystem. These protons are then trapped by the primary electron acceptor. This photosystem then replaces the lost electrons by extracting new ones from water. Okay, this is why water is important for plants as well. In this step, that releases oxygen during photosynthesis, right? So this is where we get our oxygen from, our atmospheric oxygen. Energize electrons from the first photosystem, then pass them down an electron transport chain to a second photosystem. The chloroplast uses the energy released from this electron fall to make ATP, or energy, right? The second photosystem transfers the light excited electrons to NADP plus, converting to NADPH, right? So here we have the systems showing working, right? So think of this process as the electron transport chain in the mitochondria, right? We're using this to make some energy, okay? But this is going to be the reverse reaction to what you would see as well in the electron transport chain, right? Remember the electron transport chain, oxygen is used to pull the electrons, right? Here we have water being broken apart to make oxygen in order to gain the electrons, okay? So here we have an electrical chemical gradient. Think of this as the exact same process as what you would see in the mitochondria. It's just almost reversed, right? You have the pulling of electrons from H2O, you have oxygen being made, and then you have the electrons being passed between cytochromes and these membrane proteins that allow for this proton gradient to form, right? And then you can pump these hydrogens through this enzyme in order to make ATP, okay? Those electrons can then be donated to chlorophyll, or the reaction center 2. I think this is chlorophyll A. I think it was chlorophyll B and chlorophyll A, if I'm not mistaken. This can then be re-excited by light energy, right? This photosystem or reaction center can then be excited again by the light. You have the electron jump up. These electrons can then be donated to NADP, and then this is converted to NADPH. So I just want to make a note that NADP plus is different than NAD, okay? These are 2 very different molecules, and I'll probably reiterate this again when we pick up lecture on Thursday, or sorry, on Wednesday. But this molecule again is going to be used to build glucose, right? This is not used for electrons to donate to an electron transport chain. This is used to build organic molecules. This is a building block. So mammals actually do have NADPH, and bacteria as well. They use NADPH, but again, it's going to be used as a building molecule. We use these electrons on this molecule in order to build up organic molecules, anabolic growth. Okay. Yeah, we'll talk about this next time. It's not much, but we'll finish it off next time. All right. So I do apologize about last week. I was incredibly sick, but your exam is up now. You do have that review that I did, or I posted already from Tuesday. that is still valid for this exam. If you have any questions, please let me know. I have an assignment posted already for the lab, this photosynthesis lab. I'm still working on the question. We were supposed to start cellular, oops, my internet connection's unstable. Cellular reproduction and patterns of inheritance. But don't worry, I think we'll be fine. I think we'll catch up fairly quickly. So we'll do chapter 8 and see if we can jump into chapter 9 this week as well. You do have a quiz, and I think I'm just going to quiz you on cellular respiration, cell structure, or yeah, it's just mainly chapter 6 and probably a little bit of chapter 7 too, for this quiz. But and then we'll figure out what to do with the labs. I think I'm just going to give you the same amount of labs, but I'll just add couple extra points to each so you can even out. There is an total of like 15 extra credit points too for the labs that I built in already. So don't worry. If you think that I'm shorting you points, I've already incorporated like 15 extra credit points for this kind of scenario if we got behind or if something happened. So yeah, again, this is my fault for not posting those earlier, but again, I was completely beat, so I really couldn't. It was so bad. I had a fever. I hadn't had a fever in such a long time. So but again, if you have any questions about the exam, please get it done by Tuesday. Okay, so it is due Tuesday. And then I'll kind of run through the labs in a minute. Well, I'll run through the labs right now. That way we have one recording. Let me see. Photosynthesis lab. All right, so this is going to be talking about the gas exchange. Okay, so just read over. I don't think it's very long. So this is going to kind of be checking. So I'll just tell you about the lab. So this is going to be referring to mainly how the plant can well, I guess it's more mainly referring to like CO2 dissolved in the environment and changing the pH, right? So I talked about before you can acidify your environment if you take hang on, am I screen sharing? Oh no, I'm not screen sharing. Sorry. It doesn't even tell me if I'm not screen sharing. So mainly, this experiment's going to be referring to how you can acidify the environment with CO2, among other things, right? So this phenol red is going to be, let me see if it actually, oops. So yeah, this is mainly going to be kind of showing how photosynthesis works in terms of the light and the uptake of the gas, right? So in this, I believe the water is going to have dissolved CO2 in it. The CO2 will be acidifying the environment, right? So carbonic acid. Again, if you blow a straw into water, you can dissolve CO2, and that'll provide a more acidic pH. Okay, you'll have this plant, I think it's Elodea, growing in that water. You shine the light, you'll see the pH change as this plant will uptake the CO2 in the environment. Okay, so you'll see the loss of, or I guess increasing the pH, right? So making the water more basic by taking the CO2 out of the water, so less acidic. And you'll see it with the color change of the phenol red, okay? So that's the gist of the experiment, okay? And you need to understand that plants pull in CO2 and give off oxygen, okay? And that's pretty much it, that overview for that. So, all right, does anyone have any questions? I know it's been kind of a weird last week. Any questions about the exam? Any issues? Any problems with the labs? All good? All right. Thank you so much, everyone. Again, thank you for being patient. I do apologize. Don't worry, the semester's long, so we'll get caught up. So yeah, you had a little vacation there last week for a couple of days, huh? Not just Monday. But if not, I'll upload this recording later for those of you catching this at a different time. And I'll see everyone Wednesday, or if you show up for office hours tomorrow. Okay? Alrighty. I'm gonna go and end the meeting now.